need to hit back.
It's a check, you know,
Yeah.
so I know what I'm doing here. All right, hi everyone. My name is Rebecca. I am a PhD student at the University of Utah. And today I'm going to talk a little bit about one of my studies that's looking at water use in Russian olive and comparing it to Fremont cottonwood. So to start, we likely all recognize this tree. We've been talking about it quite frequently today. This is Russian olive, which is a woody invasive tree that's really kind of escaped cultivation and now spreads pretty rapidly throughout the landscape. And what's important, Russian olive competes pretty directly with our native species, Fremont cottonwood. And Fremont cottonwood right now is in a pretty fragile state. What you're seeing here is a picture of a cottonwood stand in Arizona. What occurred here is likely a drought event where the water table drew down. Cottonwoods are highly dependent on the water table, and if they lose that connection, they can face pretty massive mortality. And so in this case, you're seeing maybe one individual out of the entire stand survived. And so when you have competition and additional species like Russian olive utilizing water, these events can be really exacerbated. And so oftentimes we do pretty extreme remediation efforts. You've seen some of those talked about today, where large stands of Russian olive have been removed off the Escalante. And the hope with removal is to release some of this water into the catchment, as well as to restore some of those shifting hydrological sort of features that we've talked about. And what you're seeing here is 3 different sources talking about the water use that we believe that Russian olive uses. So you're seeing 75 gallons of water per day per tree, 150 gallons of water per day per tree, 200 gallons of water per day per tree. And that's a lot of water, firstly. But also what I hope you can appreciate here is this is some pretty extreme variance. You're talking 75 gallons up to 200. And the reason that you're seeing that variance is because water use in Russian olive actually is not established anywhere in the primary literature. These numbers are kind of word of mouth, back of the pocket calculations. As a plant physiologist, we like to look at movement of water through the tree and scale up, and there's no primary literature that provides that sort of basis with Russian olive. And so my dissertation, one of its aims is to lay that groundwork foundation of looking at water use in this species, as well as looking at it in comparison to Fremont cottonwood. This is important to focus also on Fremont cottonwood because I just talked about that mortality event where they are starting to face really high mortality risk, but also from a land management perspective, I want to give numbers and ideas and be able to provide land managers a sort of guidepost of how much water will be released into the catchment when they remove Russian olive. But oftentimes when you remove these invasive species, we obviously hope for and want native vegetation to move back in. And so while providing numbers on Russian olive is important, having it alongside Fremont cottonwood is important as well, because it allows you to predict in year 2, 3, 4, 5, as you're seeing the cottonwoods revegetate, how that water release might change. And so in this talk, I'm going to focus on 2 specific questions. The first one is how much water does each species use at the tree and stand level? We're going to talk a little bit about that today, but also it should note that all of this data are preliminary data. I'm in year 1 of a 2-year study. And so we're going to be talking at certain scales, and just keep in mind that if I come back in another year, hopefully we'll have a more complete picture. And then the second question that I'm really interested in is how does this water use change in the face of stress? So by stress, I mean drought, heat. This is really important when we start thinking about climate change and which of these species will survive and thrive as we continue to warm. So to orient you where I'm working on the Paria River. You can see on the left, those are my field sites. There are 3 of them, and they span an elevation gradient. We like to have elevation gradients because they serve as temperature controls. I'm not going to get too far into the weeds with that today, but if you're interested in it, we can talk after the talk is over. And so I have 3 sites that I'm taking these measurements at. I should note that site 2 has some additional measurements that I will call out. They are pretty intensive, and so because of time management, they're only taken at site 2, the middle site, and I think you'll see why in a little bit. So how do we measure plant hydraulics? Plant hydraulics, basically that's just the study of the vessels that move water from the roots up through the tree and out to the leaves. And we measure this in a few different ways. I'm going to walk you through the 2 methods that I'm focusing on this talk and provide results at the same time. So we're going to go pretty quick, but hopefully it's a nice introduction to plant physiology, as well as some cool results for everybody to look at. So as a reminder, stomata are kind of the breathing apparatus of a plant. They are leaves, or on leaves, you'll find these tiny holes that open and shut. They have to be open for photosynthesis to occur, but that pathway that opens up allows water to be released through the leaf. So there's a trade-off here for plants. In order to do photosynthesis, you have to be losing water. Plants can sometimes control and regulate using these stomata. And so when we're talking about water use through a tree, one measurement that we can take is called sap flow. So what you're seeing on the left-hand picture is a sap flow installation that's installed in a Russian olive tree on the Prairie. And what this picture probably doesn't really get across is that the scale of these sensors are super tiny. So I'm talking about 1 to 2 centimeters of a very small hypodermic needle. So it's not super invasive, they're all removable. This is not a permanent installation, but it takes a lot of time and management to set up, which is why this is the one that is only installed at site 2. So this is a single site, but there's 12 trees that are rigged up at each site so that we can get variants and understand what's happening in both species. What these sensors do, they're 2 thermocouples that are inserted into the tree's xylem, and those thermocouples measure a temperature differential. That temperature differential allows me on the back end to calculate how fast water is moving. So we can know grams per hour how much water is moving through that tree continuously. Once again, I want to call out this is preliminary data. So we currently are existing, and what I'm going to present today is at the point measurement scale, all the way on the left. So that's going to tell you that instantaneous measurement of those 2 sensors, how much water is moving. But trees are all slightly different, right? We know the difference between a Russian olive and a cottonwood. One is very shrubby, multi-stemmed. One is usually a couple stems, much larger in diameter. And so that means when we take these point measurements and upscale them to the individual tree, there can be differences between the water use because of the amount of sapwood, which is the active conducting tissue that each tree has. And then again, once you scale up to the stand level, that can change again because you could have more Russian olives in the stand than cottonwoods, which means that when you look at the stand, it might be that one tree uses more water, but that when you go up another level, that might shift because there's just more of one species. So today we're only talking about point measurements. Next year, hopefully I'll come back and tell you about stand level as well. So sap flow graphs look like the following. In all of my talk, you're going to see green represent cottonwood and black or gray represent Russian olive. And so on your y-axis, you have a measurement of sap flow. Once again, that's grams of water per meter per hour, meter squared per hour. And how we look at these measurements, sap flow is continuous. So I leave those sensors in for 2 years. And it's logging that flow, that amount of water every 15 minutes for the entire day for the entire 2 years, which is another reason we're only doing this at one site. It's an insane amount of data. But so what I do on the back end is I get a monthly average of that sap flow and I map it on a diurnal sort of trend. So what you're seeing on the x-axis is time of day in June. So you can see at midnight, there's no photosynthesis. The sun is out. So there's no water movement happening in that tree. Everything's at 0. But then around 6 or 7, when the sun comes up and the tree starts photosynthesizing, you see that that water starts to move through the tree and increases as the tree begins to kick on photosynthesis. Now, as you move towards 12, 1 p.m., 2 p.m., this is the period of the day that's really, really hot. And because of that heat, trees start to shut down in order to minimize water loss. And so that's why you're seeing that decrease during the days, that stomata start to shut, photosynthesis starts to decline, and the tree conserves water. What I want to point out in this graph in particular before we move on is that at the point measurement scale, we're seeing that cottonwood uses a lot more water when compared to Russian olive. Once again, this may change as we scale, but it is a notable difference. And when we zoom out and include the data for all months that I've analyzed, this scale, or this difference is seen across all months. There's a much higher water use on the point measurement scale by cottonwood than there is for Russian olive. The other thing that's interesting to note here to me is that you can see in July and August, the months that are the hottest in our measurement period, that cottonwood has a pretty drastic shift in sap flow around the 12 to 1 p.m. period. And partially Russian olive, this is due to scale that you're not seeing it quite as dramatically. We're just looking at the curve at a more zoomed out effect, but it's seeing the same thing where there is a decline, but that decline isn't as drastic, even if you zoomed that smaller sort of Russian olive line in closer. And so if we apply this to my first question, you can see that Russian olive utilizes less water compared to cottonwood at the current scale of measurement. But I do want to continue to note that upscaling is needed, and we will hopefully talk about that in the future. So my second question looks at how sap flow varies for environmental conditions. I'm curious in looking at how these species respond to stress. So what you're seeing on the x-axis now is vapor pressure deficit, which we call VPD for short. What vapor pressure deficit is basically the combination of heat plus moisture in the air. And what's important to note here is that as your VPD gets higher, so 4, 5, 6, that means that your air is hotter and drier. As you remember, I was talking about how stomata open a pathway. And so as water travels from the roots up through the tree, that speed and that tension from the air increases as the air gets drier. And so plants get more stressed at 4, 5, 6 because the air is hotter and drier, and that pull on the water through the xylem is greater. And so some plants choose to conserve water by shutting down stomata. And what we're seeing here is a pretty different response to vapor pressure deficit. So once again, on the y-axis, you have sap flow, just how much water is moving. And you can see that as cottonwood experiences vapor pressure deficits of around 3 or 4, they begin to shut stomata and stop moving water. We don't see this as markedly in Russian olive, meaning that they're continuing to do photosynthesis even at hot temperatures at dry environments. They are not shutting down as rapidly as our natives are. And so that starts to get at my second question: how does water use in these species respond to changes in stress? When cottonwoods are faced with hot and dry environments, they stop photosynthesizing, stop using water, whereas Russian olive continue to photosynthesize and use water. This could have implications as we have more and more hot days that the Russian olive might be the one that starts using water more and more often and more frequently. Now I want to move into our second measurement type, which is leaf water potential. Leaf water potential is a measure of basically what I was talking about, that tension. So we have a scale that we've set as plant physiologists where the leaf water potential of water itself is 0. So that's the wettest you can be. The closer you move towards 0, the more and more wet you are. The more negative you get, the drier you are. So in this example, on the scale, you can see that the atmosphere is negative 100 megapascals. That means that the air is really, really dry in comparison to the soil that's closer to 0, which means it's wet. And so we take this measurement on the leaf scale. And what we're really looking at here through all of that is basically just the more negative the leaf water potential is, the more water stress that that plant is seeing. And once again, plants can control this by closing stomata. So if they don't want to risk having that water be pulled, they can shut stomata in order to conserve water and have a less negative water potential. And this also varies through the day. So this is just to walk you through where we take these measurements. This is not real data. This is just to kind of visualize. So at midnight, once again, when the sun is not up, when the air is the wettest, and your difference between the atmosphere and the soil is the least, you will see a leaf water potential that's pretty close to 0, if not 0. And we take this measurement, it's called pre-dawn measurement, because it gives us an indication of whether or not the system is droughted. So I'm going to talk you through our pre-dawn measurements as well as your midday measurements. So as the sun comes up, photosynthesis begins, that tug on the xylem becomes greater, that leaf water potential gets more and more negative, which is what you're seeing in the middle of the graph. And we take that measurement right around 1 to 2 p.m. because it's the most extreme. And it gives us an indication of what sort of strategy the plant is using, whether it's keeping stomata open or closed, as well as telling us how stressed that plant is. So I also will show you the midday water potentials for both species. So here are the pre-dawn water potentials. And really what I want you to take away is that All of these box plots, so green once again is Cottonwood, gray is Russian olive, and we've taken June, August, and September readings. In all cases, these species for the pre-dawn water potentials are below -0.5, which indicates that all of these plants are seeing drought stress. There's likely not enough water in the system for them to be comfortable, if you want to say. And so these plants are stressed in these months. In the upcoming year, we're going to expand this to include multiple months so that we can kind of see that seasonal variation. But you can see that there already is some seasonal variation when it comes to the statistical differences between months. Now, midday water potential. So I hope what you can appreciate here already, before I start walking through the data, is that there are likely two very different strategies that are occurring between these two species. On the left, you have Cottonwood, and you can see that there is a much less negative water potential in this species. They like to conserve their water potential to be around -2, if not slightly more positive than -2. This indicates, once again, they're shutting stomata. They don't want to risk that pull on their xylem. And so to conserve their health and not risk mortality, they're all shutting down, conserving water, and kind of pausing when the temperature gets too extreme. Russian olive seems to take a different tactic. Almost every month that we measured, leaf water potentials were much more negative than -2. And the other interesting thing to note is it's varying with the month. So you can see that these months are statistically different. August was the driest month that we measured. And you can see that leaf water potential is the most negative in August, indicating that the species is willing to keep its stomata open, willing to risk a much more negative leaf water potential, which puts it at risk for embolism and hydraulic failure. And so these are two different strategies, with once again this data supporting that Russian olive is going to continue photosynthesizing even in hot and dry environments. And this continues to answer my second question. So water potential in Cottonwood is conserved, which matches sap flow. Once again, we're seeing as the temperature dries that this species is shutting stomata and shutting down water use, which is the opposite of what we're seeing in Russian olive, where their leaf water potentials vary no matter what the climate is, and that they're less sensitive to that vapor pressure deficit in their sap flow. So, like I said, this is all preliminary. I think there are some very interesting findings, especially when it comes to predicting what's happening in the future with climate change. But I'm hoping next year we're starting to take those measurements to allow me to upscale. We just did some stand composition work to look at how many of each tree were in our stand. So hopefully next year I'll come back and I'll tell you a little bit about how this breaks down from the point measurement to the per tree to per stand, and really get at, when you start to remove Russian olive, how much water are you really saving, and what does that look like when revegetation occurs? We're also hoping to do some soil moisture and isotope analysis just to get a better understanding of the system, as well as understand whether or not these two tree species are using the same water sources. And with that, I would just love to acknowledge everybody on the slide. That includes my advisor, my committee, as well as all of the lovely people in my lab who have helped me out on this project. Thank you.